Well, hello there once again, BIOS 115 students. This is your instructor, Karen Klein. I hope you are doing well. Today's lab is about mitosis and meiosis. As you see from your hand, oh, hey, the dogs are upset at one another. As you see from your handout that you have, I trust you have downloaded, it's a lengthy handout. And the reason for that is because cell division, that is mitosis, that's cell division in somatic cells or body cells in order to make new ones, like maybe in growth or for repair of damaged cells or to replace old worn out cells. And meiosis, which is the division of a cell in the formation of eggs and sperm, is it's a big topic for the week. In fact, it's essential in understanding genetics, but just in understanding biology in general, and that is how cells divide. And so the first 5 pages are just review on those particular processes, but you have a lot of this in your text and in your homework questions. So I won't go over those steps one at a time, but as you know, you have prophase, metaphase, anaphase, and telophase, and mitosis, and then there's interphase when you're not dividing as a cell. And then you have meiosis. You have meiosis 1 and meiosis 2. In mitosis, the end result of when you have what we call the mother cell divide are 2 identical daughter cells, which you better hope so, because if you have a skin cell that is dividing to replace out an old worn out skin cell or a damaged one, you'd better have skin cells as a result from that one that divided. But in meiosis, when you're forming eggs and sperm, from that one mother cell, you wind up with 4 daughter cells. And they're genetically different from one another through various mechanisms, as I'm sure that you have looked at. Not all sperm are the same and not all eggs are the same, because if they were genetically identical, all the sperm that your dad makes and all the eggs that your mother makes, if you have siblings, you would all be identical. So you, of course, have variation in the 4 daughter cells, but at the end of meiosis. So what this is in a wet lab on campus is we look at onion cells at the tip of an onion, because at the tip of an onion is where you have a lot of growth. And that is where if we arrest the cells during their mitotic phases, I should say, and then we dye them, we could actually see what mitosis looks like, which is pretty cool, because it's like one thing to see mitosis on a drawing, but it's another thing to see what it actually looks like in real cells. And what you're looking for, what you'd be looking for on a real microscope slide, it looks just like this picture, is in interphase, the chromosomes aren't condensed yet into their individual little chromosomes. So what that appears like in real life is it just looks like a kind of a granular colored nucleus, as you would call. The chromosomes are in the nucleus. And so like you see one here and you see one here, these are cells that are in interphase. Now prophase, as you recall, you have, let's see if can we get bigger here? Oh, we can. Oh, hot dog. You know what? Great. That is so much better. So, well, here's interphase. You see how it looks granular, but you don't see any individual chromosomes. Now, in prophase is where the chromatin, condenses into visible chromosomes, but the chromosomes haven't really lined up on that metaphase plate yet. So it just looks like a plate of spaghetti. the chromosomes look like spaghetti noodles. So you see here, these are the stained nuclei. Here's prophase. Oh shoot, no, there's interphase. Prophase, I said that right, is where they are condensed, but they haven't really configured themselves in any order. So like here's a good prophase. Here's a good prophase. Now in metaphase, those chromosomes line up along the metaphase plate. And so what it looks like, I mean, in one of those drawings, it's very tidy, it's very neat, but nature is never that tidy and neat. So we have to kind of, we have to look for where it looks like actually those spaghetti noodles are just like slammed together in the middle, middle of like a dinner plate. So where can we find an example? Oh, there's a good one right here. You can see where those chromosomes, here is the cell, and they're kind of like smooshed together in the middle at the equator of the cell. So there's a good depiction of metaphase. Again, here's one too, right next to it. Looks kind of sloppy, right? Well, like I said, it's nature isn't as tidy. Now, anaphase is where, chromosomes are made up of sister chromatids, 2 sister chromatids. And in anaphase, they pull apart from one another. Chromosomes are made up of 2 strands, sister chromatids, and they pull apart from one another. And so what it looks like is right here. You see how they pull apart from one another? You could see that it's like somebody, again, took those spaghetti noodles and like separated them. So here's a typical anaphase. And telophase on a microscope slide is where you see a cell that's, or 2 cells that are half the size of a regular cell. The chromosomes have pulled apart from one another, but the cells, the 2 new daughter cells, are still pretty small, and they haven't really formed 2 cells yet. That is going to be here. This actually, this is kind of a late telophase. I'm sorry, a late anaphase. You can find late anaphase, early telophase, because you can see that they've clearly pulled apart from one another, but you can still see the chromosomes, because in later in telophase, you don't see the chromosomes at all anymore, and it's just these 2 that have occupied. Like, yeah, this is like a later telophase. You see, like here is 1 cell. You see how big that is, and now here it's the size of a 1 cell, but it is just the 2 smaller ones. That indicates that telophase has taken place, and you're almost, then you go back into interphase again, where they will grow to be the size of a regular cell. Now, for your exercise, though, I couldn't really do those screenshots, and so what I did is I took off of Google Image, I took an onion root tip under magnification, and I have on the last page pointed out A, B, C, D, and E, and I have the pointers there. And so what I want you to do is identify which phase of mitosis that represents. Well, whether it's interphase or a phase of mitosis. So you want to put that into your, right on the sheet, and then turn that in for this week's lab points. Okay, everybody. Hope all is well, and we'll see you later. Take care. Bye.